Hello everybody, welcome back to my laboratory where safety is #1 priority. Today we'll be doing the Cricket Cellular Respiration Lab. The problem we'll be testing is how temperature affects the metabolic rate of a cricket. In order to measure this metabolic rate, we'll be using a respirometer. The respirometer is a device consisting of a base chamber and a tube. The test subject will be placed in the chamber on non-absorbent cotton with absorbent cotton containing potassium hydroxide beneath it. The tube connected to the chamber will be filled with a small amount of food dye. As the test subject breathes, the food dye will be sucked down the tube, and this distance will be measured as cell respiration rate. In order to prevent the cricket from being poisoned, potassium hydroxide in the absorbent cotton combines with carbon dioxide to form a solid precipitate. The temperature variables being tested will be hot, 20 degrees Celsius, and cold, 10 degrees Celsius water, and their effects on the cricket will be measured.
So to sum up, the problem is how does temperature affect the metabolic rate of a cricket? The independent variable is the temperature of the water, either 10 or 20 degrees Celsius. The dependent variable is the distance that the food dye has traveled. The control group is the non-living crickets, as opposed to the living crickets. The controlled variables are the chambers, washers, tubs of water, available light, food dye, amount and type of cotton used, and the amount of potassium hydroxide used. And the hypothesis is that the living crickets in the warm water will have the highest rate of respiration because their metabolic rate will be the highest as a result of being in the warm water because they are cold-blooded creatures. Appearing on the screen now is the results for the respirometer in either warm or cold water. Obviously, there would be two finished data tables at the end of the experiment. It shows how, over time, the fluid moved through the tube in both the control and the experimental group. It is also worth mentioning that the average rate of travel for each trial of the fluid through the tube should be calculated. This was done by us in centimeters per minute.
Now we will set up the control group. We will take a fake rubber cricket and insert it into the tube.
Try and make sure it's the same size as well.
You can also use this part to help shove it down if you need to. Master skills. Now, to insert the cricket within the tube, you just kind of want to not drop it on the floor.
Don't break the legs either.
Yes, and try not to kill it. It may stick to the top of your hand, like that. Just slide it off, and it should drop. Or you might occasionally rip their leg off, but oh well. They don't really as we just show that one of their legs is ripped off. Now we shall put the stoppers on top of these also.
Now we'll have all 4 tubes complete, 2 with live crickets and 2 with the control crickets.
We put ice in this one to make it colder.
Hot water to make this 20 to 25 degrees Celsius. Now we'll insert each into the solution where the environment is. And allow them to adjust to the environment temporarily to allow the air temperature to change.
For around 3 minutes. Okay, now that 3 minutes have passed, we will start to weight down each tube. We shall slide washers, preferably steel, down the tube on top of each canister that holds the crickets. Preferably do this before you add tape to the top.
Sorry, I didn't hear you. I was trying to figure it out, and then I got it. Absorbent cotton. Yes.
Add as much weight until they sit perfectly down. The tape will help keep the tubes from moving side to side. We shall do the cold water blue and the warm water red. Line up the food coloring with the top of the tube and slightly press. Now we shall do the warm water with the red food coloring. Oh. Try not to let it touch your skin because it will stain. This is at 9, we will try to get it to. Get it to at least, this one will be kept at 10 degrees Celsius, while this one is 20 to 25 degrees Celsius. The start time we have started is at 1:14, and the experiment will last for 25 minutes. So now we shall cut to the time lapse. Have concluded this experiment. As you can see, the cold water has gone down little to none. Probably actually none.
And the non-living specimen.
Yep. But as you can see, the warm water has had a drastic increase. Even the non-living, the plastic cricket, has gone down.
Our data table for the warm water, or 20 degrees Celsius water, is shown here. It also shows the average rate of travel of the fluid down the tube. Please note that this rate is calculated based on the initial value at 0 minutes and the final value after 25 minutes. My group's results for the cold water, or 10 degrees Celsius water, are shown here. Also, the average rate of travel of the food dye down the respirometer tube is shown. All of these results will be explained later in the conclusion.
The variation in food dye travel can be explained by different rates of cellular respiration. As temperature increased, metabolic activity, which includes cellular respiration, also increased, while as temperature decreased, metabolic activity also decreased. This characteristic is unique to cold-blooded organisms such as the crickets we observe today. This explanation is supported by the results of the data table. After 25 minutes, the live cricket in the warm water's food dye traveled 4.8 centimeters, while the living cricket in the cold water's food dye only traveled 0.2 centimeters. Although this is true, temperature also had an effect beyond metabolism. In the warm water, the plastic cricket had a 1.8 centimeter travel in its food dye, while the plastic cricket of the cold had a 0 centimeter travel. Changes in temperature also cause a change in pressure, which is responsible for the pushing effect found in the non-living crickets.